So, welcome to Biomindscape. Today, we are going to know about the amino acid metabolism. In this, we will know amino acid synthesis, where the living organism have a different capacity to synthesize amino acid, which is important for the protein synthesis for making the protein synthesis. So, while the plants and animal microorganism they produce 20 standard amino acid, which are available as a precursor, and other organism obtain it through the diet by an amino acid diet. So the mammals and other animals they can synthesize a substance of amino acid where we have 12 non-essential and the remaining 8 non-essential amino acids. So let's name it: isoleucine, leucine, lysine, methionine, phenylalanine, then threonine, tryptophan, valine, arginine and histidine. These are essential amino acid, and non-essential amino acid are alanine. Here alanine is non-essential one. Then proline, asparagine, aspartate, cysteine, glutamate, glutamine, glycine, serine, threonine. These are the non-essential one. Then the nitrogen is important component of an amino acid, which form an ammonium group as a source of nitrogen, which is for all the amino acid. So it incorporates in amino acid into various other biomolecule like nucleotide, and the two main amino acid like glutamate and glutamine, it provide an entry point. Like glutamate, it is a source of amino group for most of the amino acid through transamination reaction. So, two important principles by which amino ions are incorporated into amino acid are eventually other metabolisms. So, here other metabolites. So, the reductive amination of alpha ketogalic acid, where alpha ketogulate, if we have taken an alpha ketogulate and we have added NAD H your group here, and it is reversible reaction where it forms the glutamate. Why here what happens? The glutamate dehydrogenate catalyze the reductive amination of alpha ketogulate and it form the glutamate. So this pathway is present in all the organism, and in eukaryotes it is the minor pathway and it occur at a specific cell organelle, that is a mitochondria. So glutamate dehydrogenase is unusual enzyme and it does not discriminate between NADH and NADPH. Then there is a formation of amide of glutamic acid, where the glutamate is getting converted into glutamine when we add ATP. So amino ion is present here in this cell metabolite. It form a glutamine. So the amide of glutamate, here this amidation reaction, it is catalyzed by glutamine synthase, which is present in all the organism, and it is derived by hydrolysis of ATP. So here there is a participation of ATP which helps in the reaction by phosphorylating the side chain of glutamate, and it form acyl phosphate intermediate, and after further reaction it form the with ammonia it form glutamine. So, for example, glutamine, if we add it with alpha ketogulate and the reactant is NADH, so at that time we will get glutamate. So, in plants, after ammonia is incorporated into glutamine, the glutamine synthesis happens. The amide amino group is transferred to 2-keto group, that is of alpha ketogulate by glutamate synthesis. It is also known as the glutamine or 2-oxyglutarate aminotransferase or GOGAT. So this glutamate synthesis is transferred to the amino group of glutamine to alpha ketogalic acid, and it forms a 2 molecule of glutamate, that is amination of alpha ketoglutarate. Now let's know what is a transamination. So many amino acid biosynthesis pathway are involved in this particular transamination reaction, where the alpha amino group it form one amino acid and it is transferred to alpha keto acid and it produce a new amino acid. So this reaction is catalyzed by an enzyme called aminotransferase. It is also known as transaminase. So aminotransferase, it requires aldehyde containing coenzyme. like the pyridoxal 5-phosphate, and it is derived for vitamin B. So this pyridoxal 5-phosphate is the it is attached to an enzyme via skiff base linkage, which is formed by the condensation of aldehyde group with pyridoxal. It is a part of the coenzyme which helps in generating the pyridoxamine phosphate. So the pyridoxamine, it is form of one coenzyme. It reacts with alpha keto acid and it forms an amino acid. So main what happens, these pyridoxal 5-phosphate, it reacts with alpha keto acid to form an amino acid and regenerate the original aldehyde form of coenzyme. So eukaryotic cell have variety of amino transferase. It have a large variety. So it is found in both the cytoplasm as well as the mitochondria, and this enzyme poses two type of specificities. They are alpha amino, which donate the alpha amino group, and alpha keto acid, which accept an alpha amino group. So aminotransferase have a variety wide range of amino acid that binds, and most of them are glutamate as an amino group donor. So the amino group from the glutamate, it can be transferred to alpha keto acid by transamination reaction. So transamino reaction, it helps in synthesizing of most of the amino acids. So there is an acceptor alpha keto acid, then glutamate. Here we add the glutamate, so it is a reversible step where the new amino acid is formed plus the alpha keto glutarate. Then let's know about the carbon source. So we are going to know about an overview of amino acid biosynthesis, the carbon skeleton precursor, and they are derived from a three source, that is the glycolysis TCA cycle and pentose phosphate pathway. So here there are various families of amino acid. So these family together they derive for a particular one precursor molecule. So first is the glutamate family. It includes the glutamate, glutamine, proline and arginine, and they derive their carbon skeleton from alpha ketoglutarate. Then serine family, these are the member of serine family. Here serine, glycine and cysteine, they are derived their carbon skeleton from glycerate 3-phosphate. Then aspartate family, here the aspartate family include aspartate, asparagine, lysine, methionine and threonine, and derive from their carbon skeleton from oxaloacetate. Then there is a pyruvate family. Here the pyruvate family, it consists of alanine, valine, leucine, isoleucine, and they are derived from the carbon skeleton from pyruvate. Then aromatic family, where it is derived from the phosphoenol pyruvate and erythrose 4-phosphate. So here the members of the family are phenylalanine, tyrosine, tryptophan. These are the members. Then histidine. Histidine is derived from a carbon skeleton from ribose 5-phosphate. So here we have seen the diagrammatic representation of the overview of amino acid. Thanks for watching. Do like, share and subscribe.